There are MET-addicted subtypes of non-small cell lung cancer. High-level MET amplification, but perhaps more prominently MET exon 14 skip mutations, which is about 4% of lung cancer. There's a range of tyrosine kinase inhibitors, most of them are what's called class 1 inhibitors, crizotinib, tepotinib, and the most recently FDA-approved one, which is capmatinib. There are also some class 2 inhibitors, but none of those are actually licensed. Cabozantinib is sitting in the pharmacy, although it's a relatively poorly tolerated drug. Now, these drugs are good, but you all develop acquired resistance. And some of those resistance mechanisms are on target. You get MET mutations. You can sometimes change between class 1 to class 2, but another approach is whether you attack MET with an antibody. Sym015 is a mixture of 2 different MET antibodies. And although, yes, it shows great activity in those who are untreated with a MET inhibitor, as you might expect, who's going to go for an intravenous drug as opposed to a pill? But what's exciting about this presentation is a relatively small number of cases of patients who had been previously exposed to a MET tyrosine kinase inhibitor had developed acquired resistance, including at least one who had definable MET mutations. And you could show that their cancer shrunk. It didn't quite achieve an objective response, but you could show that those mutations went down in circulating free DNA. And so these antibodies may have a role either in the acquired resistance setting or possibly in combination with the tyrosine kinase inhibitors up front.